Hello, everybody. Welcome to this session, to day 2. We have 3 speakers lined up for this session. I am Shaychuti. I am co-chairing this session with Shyam Prabhakar. I specifically focus on, for my career, focus on microbial genomics and pathogenesis. I'm very excited to be the chair of this session. I think I'm going to hand over the microphone to Shyam to introduce himself.
Thank you, Shaychuti. Shyam Prabhakar from the Genome Institute of Singapore. My group does algorithms and disease mechanisms, and looking forward to an exciting session on genetic diversity, which is really very close to the heart of Human Cell Atlas AIDA.
Okay, I will invite the first speaker. Each speaker will have 15 minutes to present their talk, and speakers, we will give you reminders when you have about 5 minutes left. And then after each speaker, we'll have 10 minutes of question answer. So our first speaker is Deepa Rajagopalan, and Deepa completed her undergraduate degree in engineering majoring in industrial biotechnology from Sastra University, India in 2013. During this time, she worked at the Indian Institute of Science between summer of 2011 and 13 and University of Michigan, Ann Arbor. She moved to Singapore to pursue her PhD in the National University of Singapore, and her thesis was carried out under Sudhakar Jha's lab, and she identified distinct roles in licensed acetyltransferase, TIP60, and HPV-mediated cervical cancer, and in the inflammation associated with colorectal cancer. She's currently working as a postdoc with Shyam, actually, aiming to generate an atlas of immune cells from healthy Asian populations. And this will enable future studies on characterizing the immune cells in tumor microenvironments. So, Deepa, the floor is yours. Thank you.
Hey, thanks for the introduction. And let me just share my screen and get started. Hope everyone can see my screen. I'm really excited to be here. Thanks for the introduction. And I want to thank the organizers for giving me this wonderful opportunity. I'm new to the sickle cell field, as you heard, and I started only when I started in Shyam's lab. And this meeting was my first Human Cell Atlas meeting, and it's been absolutely amazing. I've learned a lot. So without further ado, let me talk more about my project in Shyam's group on developing and building the Asian Immune Diversity Atlas. So I think we all know that genetic studies, especially large-scale genomic studies of human diseases, are a bit skewed in the representation of the global diversity. Specifically, these studies have a higher representation of individuals with European ancestry. So you can see here, this is a catalog of all GWAS studies, and about 80% of the individuals represented here are individuals with European ancestry. And this is not unique to GWAS alone. Another genome aggregation database, Nomad, also has the same caveat. So about 60% of the total of 140,000 odd genomic and exomic sequences come from individuals of European ancestry. And similarly in GTEx database, again, Asians, American Indians, and African Americans are represented to a much lower extent. So we do understand that some diseases have an origin in genetic architecture, and this lack of ethnic diversity could pose problems. And while trying to understand and read more about this, I came across this fascinating commentary in Cell, which addresses the same question of missing diversity in genomic databases. So basically, if there is a Mendelian disease, which is attributed to a single gene, which has a large effect size, then the transferability of this between different population groups could be rather high. On the other hand, when you look at diseases that are caused by more than one gene with moderate effect sizes or many genes, which are basically polygenic diseases, these risk scores that are derived from here could not be easily transferred between different population groups. So basically, studies that are only derived from European individuals could not accurately predict risk in other population groups. And there is another inherent caveat here, which is that if these risk scores are assessed using tag SNPs, then it adds to the complexity because the tag SNPs may or may not be similar across populations. And this is what is highlighted here. So you could have one causative variant across all the different population groups, but it could be identified by a different tag SNP based on the different populations, right? And then if these are different, it's harder to map it back across the diverse population groups. And if you have different causative SNPs, again, that becomes complex. So basically, the underrepresentation of ethnically diverse populations could impede our ability to fully understand this genetic architecture of human disease. And this becomes particularly important when you look at diseases that do have a genetic component. And I'm just depicting two examples here, but there are a lot of studies which show this. So the example on the left here is talking about coronary artery disease and the mortality associated with it and comparing it with the total cholesterol levels. So we can see that as there is an increase in the total cholesterol, there's an associated increase with mortality, which is not surprising. It is interesting, however, to note that between the different population groups that are compared, there are inherent differences in the mortality. So between Asian Indians, who seem to be more predisposed to coronary artery disease as compared to the Japanese population, it suggests that there are probably underlying genetic, environmental, probably lifestyle-related effects. And thus it reiterates the need to have a genomic database that is representative of all of these differences. Another example with diabetes, type 2 diabetes, another common disease, but again, a complex disease because it cannot be attributed to one single particular gene. And there are differences. So this is the study from the United States, which talks about the prevalence of type 2 diabetes among different age groups. And you can see that in older individuals, American Indians or Alaskan Natives have about a 45% prevalence, whereas this is only 17% or so in non-Hispanic white individuals. So all of this reiterates the message that there should be a stronger, larger effort to characterizing all the diverse populations. And we are trying to learn from all of these previous consortia. It is an ongoing challenge because this is a rather big problem. And at the Asian Immune Diversity Atlas, we're trying to really build a single cell representation of all the Asian immune cell types. And with this, we want to look at differences that exist within and between different Asian populations. So we're looking at samples from Singapore, which happens to be a multi-ethnic city-state country with individuals of different ethnicities. So we have Chinese, Malay, and Indian ethnic individuals here. We're also looking at samples from South Korea, Japan, India, and Thailand. So as I mentioned, this is a multinational effort and it's led by people in different countries. So Sham leads the group here in Singapore, Woong Yang in South Korea, Che in Japan, and more recently, Pata from India and Baradom from Thailand. And together we have also got a lot of other investigators who help us with sample collection, data generation, analysis. And so you can see that this is really a massive team effort and it's quite an exciting project to be working on. So what's our goal? Our goal, again, as I mentioned, is to generate this atlas of all the different Asian immune cell states and to characterize the differences, if any, within and between these Asian population. And the key thing that we're doing is using common controls, and I'll touch upon this later as well, but this is just sort of control for batch effects between the different sites. And what we really want to do is build this reference of healthy individuals for all future disease studies in this population. So our first aim is to profile blood, so single cell PBMCs from 6 different population groups, as I mentioned, and these 5 countries. And we're looking at healthy individuals. We'll do single cell RNA sequencing, single cell ATAC sequencing. And since we plan to do multiplexed assays, we would also do genotyping. And our aim is to go at least up to 500 samples in total for the pilot. We want to develop algorithms which would correct for batch effects using common controls, as I mentioned. We want to identify different immune cell types, immune cell states, and also really look at the immune repertoire on the T cells and the B cells. We want to look at differences within and between population groups and see if these could be attributed to gender, to environment, and so on. And of course, eventually we want to submit the data to DCP. And I'm happy to say that we have indeed submitted our AIDA Pilot datasets to DCP already. So it's going rather well. So the overall workflow involves collecting blood and isolating PBMCs from blood. Then we do single cell RNA sequencing, ATAC sequencing, and this would be from pooled PBMCs. This would also have a component where we do genotyping of all the same samples, and then we'd use our novel algorithms to try and uncover all these interesting biological insights. So the approach that we adopt has been pioneered by different groups, particularly Jimmy's group, and this involves using pooled samples. So we're doing MUX-seq, where we pool different individuals, unrelated individuals, in one single 10x or assay reaction. And in one droplet, we would have distinct genotypes. We would then sequence it all together. So the cDNA is labeled with the genetic and cellular barcode. And eventually, after sequencing, we demultiplex this using an algorithm, demuxlet, to remove the doublets. But it will also help us assign identity to each of these individual cells. So we demultiplex and remove the doublets using this bioinformatic algorithms. Yeah. And this is important because it now brings down the total cost, because you're dividing the cost for one single reaction amongst how many ever individuals you're pulling in. So it brings down the total cost involved as well as the library preparation cost. So for AIDA, we're pulling cells from 15 distinct Asian individuals along with one common control. So 16 individuals in one reaction. And we want to load 40,000 cells per reaction. The goal is then to recover about 18 to 20,000 cells from each reaction. And after we do our QCs and remove the doublets, we hope to have 16,000 single cells from one reaction, which would be 1,000 cells from each of the 16 individuals. So if we do 2 reactions per pool, we get 2,000 cells per individual, which is a rather good number to start with. And we will sequence these 2 reactions in one lane of the NovaSeq S4 chip, so about 2.5 billion reads and 40,000 reads from one single cell. So before I get to discussing the data, I just, for any of you who want to do or conduct such a multi-site, large-scale single-cell study, these are some of the things we've learned in setting up this project, and I wanted to share this with you all. So the first, and even though it might seem rather trivial, but the most important thing is to really find the right collaborators. So find people who are working towards the same scientific goals, who have this willingness to share their methods, to share their data freely across the sites, and of course, who have the technical capabilities to conduct the study, right? And who are willing to get on calls frequently to discuss, and there's a constant back and forth and sharing of information. The second thing that we all had to do was to standardize all our protocols and everything. I'll get to that, but to standardize questionnaires, which include inclusion-exclusion criteria for sample collection. We also wanted to make sure that the ethics are in place, so confirm that the data can be shared via the DCP and standardized sample collection protocol. And we've uploaded our protocols at protocols.io. Feel free to refer to it, and if you have any questions, you could always get back. The other thing that's significant about our study is that we're using a common control. So we have commercially available controls that we buy, and these are processed as reference samples at all sites, and they are there in every batch of our AIDA samples. We also will randomize the batch standardize the batch randomization strategy, and of course, standardize the single cell processing protocol. Again, our protocol is available at protocols.io. The next thing after we generate the data is to send it for sequencing. So we have centralized DNA sequencing and the primary data analysis, and the secondary data analysis will of course be done at each of the sites. Right. So our pipeline, and this is I work very closely with Quay, who's a computational biologist also working on AIDA, and this is what we use for our analysis. So once we get the raw sequencing data, we process it using cell ranger. So cell ranger count to get to a gene cell matrix. Then we do a loose QC basically to discard the empty droplets, and we have a filtered gene cell matrix. Here at this stage, we use 2 different algorithms to try and remove most of the doublets. So demuxlet helps to remove intra-sample doublets. Doublet finder would help to remove heterotypic doublets between samples. And we are left with demultiplexed reads, which are from singlets. And here we use an algorithm reference-based clustering, which is developed in Shyam's lab to annotate major cell types. So this is called RCA2. So we would look for most of the common cell types that are usually seen with PBMCs. Right. And after this, there's again an additional step, and this came about again from Shyam's group, where Nirmala and Joanna suggested the need to do a cell type specific QC, and I'll elaborate on this in the next 2 slides. But basically, we do a cell type specific QC, and these post QC cells are then used with Seurat data integration to integrate data from all the different AIDA sites and to start looking to see if there are any batch effects. Okay. Before we went further with the data analysis, we also wanted to make sure that our demultiplexing was, we could demultiplex samples efficiently. So we use 2 different algorithms, the demuxlet and freemuxlet. Demuxlet uses genotyping information, freemuxlet does not need it. And you can see that there is a high concordance between these two algorithms in terms of demultiplexing our library. So this is a pilot library with 4 samples, a pilot library with 16 samples, and we're able to confidently call for demultiplexing lit samples. Right. So talking about the QC metrics. So you can see that our QC standards depend on the number of detected genes as well as the percent of the mitochondrial transcripts in each cell type. And you can see that this actually varies between the different myeloid and the lymphoid cells, and also between each of the different cell types. So we cut off for each cell type, and that's indicated roughly by these red boxes, to get a cell type specific QC to obtain our high quality cells. And this data set is representative of all the cells that we have from Japan, South Korea, and Singapore. So to visualize it better, so here's how the cells look before our QC, and we can already see all the major clusters that you would expect from a PBMC data set. However, when we look closely before QC, you do see these, okay, I'm sorry, can't see my mouse moving, but yes, you see these low quality cells here, which are marked by low NODG, high P mito. And these are the cells that are filtered out, marked in red in our post after QC. So the cells that we're left with are the post QC cells, which are colored in blue. And we then lose most of these lower quality cells, which could also be some homotypic doublets that were left undetected by our two algorithms. Right. So after this, we wanted to, the important question was to see if all our standardization across all the sites paid off. Was it effective? And so we used Seurat-based data integration and projected all the data from all the sites onto the same panel. And indeed, it's the case. So we do see that between Japan, South Korea, and Singapore, the data is highly coherent, and there are very minimal batch effects, if any. And another representation of the same data set is shown here, where data from Singapore, Japan, and South Korea, the cell type proportions of all the major cell types are fairly similar across the different sites. So this is our work on the single cell RNA sequencing. We also have initiated pilot studies which look at the TCR and PCR profiles, and I'm just showing data from our Singapore dataset. So we have, again, we start with the raw FASTQ files and we use Cell Ranger, but use a different Cell Ranger program called Cell Ranger VDJ to get the contig and cell matrix, and then use a few different programs, algorithms, if I may. So SE repertoire and RCA2 to do the further clonotype analysis. And over here, you can see this is not very surprising in terms of findings. So we identify unique clonotypes across the 4 different individuals, both for TCR and for BCR. Considering that they are healthy individuals that we are looking at currently, there is no hyperexpansion of any given clonotype, and there's a fairly large percent of unique clonotypes, which should be expected for this data. And we can also see very nicely that these are distributed across the T cells and the B cells, and we have information on where these specific TCR and BCR repertoires are coming from. So in summary, what I've showed you is that the most important takeaway is that the QC statistics are certainly dependent on cell type. So doing a cell type specific QC would help us get only the highest quality cells for further downstream analysis. And so far, all our standardization protocols across the different sites in different countries have ensured that we have minimal batch effects, at least from this data set that I've just shared with you. Our plan is to scale up, as I mentioned, to have 500 samples. We want to expand also to additional populations to have a more representative Asian atlas. So feel free to reach out to us on this. We are developing algorithms to use common controls for batch normalization, and also looking at developing pipelines for profiling regulatory elements using 5' single cell RNA sequencing data. And the TCR-BCR pathway pipeline that I just showed you is also rather preliminary. So we want to characterize this diversity in further detail. All right, before I take questions, I want to take a moment to thank all of our wonderful collaborators. So people from John Chambers' team, people from Jay's team, and Raikan, Yoshi, and Jonathan, and people from Woong Yang's team in South Korea. And of course, I want to thank people from Sham's lab at GIS, Kwe, who I work very closely with for the analysis, Lamin and Prasanna, who helped me with the sample processing, Nirmala and Shweta, who were instrumental really in initiating this project, getting the study started even before I was at GIS. Okay, I'll stop here and take any questions. Thank you.
Thank you, Deepa.
Yes.
We have one question in the Q&A from Jose Hernandez.
Right.
How accurately do you think you can demultiplex 15 samples from a single pool, given the genetic relationships between the individuals?
Yeah, I think it's fairly concordant. So I haven't shown the data, but we also, I'm just trying to go back to comparing the slide which compares demuxlet and freemuxlet. That's a great question. So we did try it. So we have tried to look for both comparing with and without genotyping information, which is what I showed. There are very few that are actually ambiguous or called differently by two of these algorithms. We also compared two different genotyping based algorithms, so tmuxlet and supercell. And again, the results are fairly concordant. So I would say we are confident in calling apart these 16 individuals in our pool. Yeah, that's a great question.
I think I have a comment, Deepa.
Yes.
That was a wonderful presentation. Thank you so much. I don't have a strong background in single cell analysis, but I understood a lot of it.
Okay.
And I was also so happy to see the amount of work that is being done to understand the genetic diversity within Asia, because I mean, there's a lot of discussion about the lack of diversity. Just a couple of weeks ago, there was this comment published in Nature, how like, minority, I don't know why we are called minority, but minority data systematically excluded from such kind of analysis. So thank you and Sham for doing such an important project and explaining it so well. Thank you.
Yeah, I mean, I just have to add that this is such a huge team effort, and we have people that we're working with across Asia, and it's been a great journey so far. And I'm looking forward, now that we have everything in place, we're going to be generating a lot of data rather quickly. So I'm excited to see how it turns out. Yeah. Okay.
Deepa, we have one more question in the chat.
Okay.
From Yulia Medvedeva.
Okay.
For the genotyping, you're using a custom array. Do you expect this custom array to work in other populations?
Okay, so that's a great question. I think we're using an array specific for the Asian population, but I'm not sure what you mean by other populations, but I think it should cover most of the common SNPs in Asian data sets. And if we do need to, we might have to do some imputations if we're unable to really pass apart the different individuals. But yeah, I don't know if I've answered it accurately, but that's my take on it. Yeah.
Maybe it's also a reference to non-Asian populations.
Yeah, so then we can start look at imputations from the 1,000 genome panel. I think that there are different chips. I think the reason we chose this was because it was specific to the Asian population group. Yeah. Okay.
We have time for one or two more questions.
Yes.
While we're waiting for people to type,
yes.
I think it'll be useful for the audience to say, what were the considerations, if someone else wants to design such a multinational single cell study where you're comparing populations or ethnicities or so on.
Yes.
So.
Yeah, like right here, I think the most important, sorry, Sham. Yeah, I think the most important consideration should be this, finding the right collaborators and to really standardize protocols. At least in our hands, we can see that all the efforts that we put in to standardize questionnaires, to standardize reagents, to the extent that we order the same consumables with the same lot number, the same batch, but just distributed to the different countries. So we reserve it in advance and for the whole length of the study. So I think measures like this have really helped us overcome batch effects, which could creep into a study of this scale. Yeah.
Specifically about, what you centralize, what you what you do in all in one place, what you do at the original site, things like that.
Right. Yeah. So I had color coded it in the sense that the green things are the ones that are centralized. The dark blue is what is done at each site, but it's of course done in concert with all the other sites. So we do standardize a batch randomization, we do standardize all our protocols and coordinate on all the samples. So the common controls are also ordered from the same batch, same lot, and just distributed to the different sites, so by the distributors in each of those countries. So all of that is at every site. And then the sequencing and primary data analysis is all centralized. So we're doing it in Singapore now. So everyone sends in their DNA samples. So the samples are collected, the single cell work is done at each of these countries, and the sequencing is done in Singapore, and the primary data analysis, just the QC, and everything that I just explained to you. Of course, the secondary data analysis is again done across all sites. And there are frequent discussions on, the data and the interpretation and so on. Yeah.
Great.
Okay, I don't think there are further questions.
Thanks very much, Deepa.
Okay, thanks, Shyam. Thanks, everyone. Happy to discuss. Yeah. Let me stop.
Thanks again, Deepa.